Hi, in this video we're finishing out our senses. This is going to be part 2 of a 2-part video series. We're going to pick up with the sense of sight. Now with the sense of sight, there's going to be a number of accessory organs that's going to assist with the visual receptors in the eyes. These are going to include things such as the upper and lower eyelids, the lacrimal apparatus to help protect the eyes, and a set of extrinsic muscles that can help move them. So each eye is going to have eyelids, which are called the palpebra. It's going to be composed of 4 layers of skin. With those 4 layers of skin, it's also going to have muscle and connective tissue, as well as a conjunctiva. Now the conjunctiva is going to be a thin layer of skin that's going to be on that outer surface that's going to cover the eye as it goes across, and it's going to fuse with the inner lining of the margin of the lid. Next, you're going to have your eyebrows. With the eyebrows, it's going to help to shade the eyes. It's also going to help to prevent things like sweat from trickling into the eyes. And so basically, that's just going to be another protective mechanism to help protect them. You have your eyelashes, which are going to come in next. With the eyelashes, they are going to be very sensitive, and when they're touched, it's going to stimulate that blinking reflex. And going into that conjunctiva again, like we said, that is a transparent, very thin membrane that's going to cover the main portion of the eye. So it goes across the sclera. That's going to be the white part, and it's going to help to produce mucus. Now looking at these structures, you can see each part. You can see the eyebrow above, which is going to be the hairs that are going to go around the superior ridge that's going to be above that opening for the eye. You have your eyelid. You have the eyelashes. You can see the site where the conjunctiva is going to merge with the corner. You can see the palpebral fissure. Then you could go in and see the iris. That's going to be the colored portion of the eye. You have your eyelid on the bottom. The pupil is going to be the opening of the eye, which is going to be controlled by smooth muscles, which we'll talk about. The sclera, which is going to be covered by the conjunctiva. That's going to be the white portion of the eye. And then you have your lacrimal duct, which is going to be sweat and sebaceous glands, which are found in the medial portion of the eye. Your lacrimal apparatus is going to consist of the lacrimal gland, which is going to secrete tears, and a series of ducts, which are going to carry these tears into the nasal cavity. So if you've ever had like a really ugly cry or a bad cry, you've noticed that your nose becomes runny. That's because these tears are being directed down through ducts towards that nasal cavity. The gland that's going to be in the orbit, superior and lateral to the eye, is what is going to be producing these. So it's going to secrete tears continuously, which are going to pass through tiny tubes and flow downward and medially across the eye. 2 small ducts, superior and inferior canaliculi, are going to collect tears, and their openings, which are called puncta, can be seen on the medial borders of the eyelids. From these ducts, the fluid's going to move into the lacrimal sac. This is going to lie deep in the groove of the lacrimal bone, and then into the nasolacrimal duct, which is going to empty out into that nasal cavity. You're going to have glandular cells that are going to be found in the conjunctiva that's also going to secrete these tear-like liquid secretions together with the secretion of that lacrimal gland. So all of this is going to help to keep that eye moistened. It helps to keep it lubricated and keeps the surface of the eye lining very moist and able for you to see very well. Also helps with your eyelids as you're blinking. It helps them to be very smooth as they're going across the eye and cleaning the eye off. Next, you're going to have your extrinsic muscles. There's going to be 6 of these muscles that are going to help to rotate the eye in various directions. And so you'll have the superior rectus that's going to rotate the eye upward and toward the midline. You're going to have the inferior rectus that helps to rotate the eye downward and toward the midline. You have the medial rectus that's going to help to rotate the eye towards the midline. The lateral rectus, which rotates the eye away from the midline. The superior oblique, which rotates the eye downward and away from the midline, and the inferior oblique. And this is going to rotate the eye upward and away from the midline. So the motor units of the extrinsic eye muscles are going to have the fewest muscle fibers, only about 5 to 10 of them. And that's going to be fewer than any of the other muscles of your body. So it's going to enable them to move the eyes with great precision. Also, the eyes together helps put them together so that they can align when you're looking at something. So it helps you follow something as you're trying to watch it. This alignment is going to result in complex motor adjustments that can contract the eye muscles while relaxing the antagonist muscles. So, for example, when the eyes move to the right, the lateral rectus of the right eye and the medial rectus of that left eye are going to have to contract. And so at that same time, that medial rectus of the right eye and the lateral rectus of the left eye are going to have to relax. And that's going to allow these eyes to move together. So they're going to have to kind of be like opposites. And so looking here, you can see that lacrimal gland, which is going to be found on that lateral portion of the eyelid. So that's what's going to be secreting the tears. They're going to come out through those excretory ducts of the lacrimal glands, and they're going to work their way across the eye to keep it lubricated, to keep it moistened. And as they work their way across the eye medially, they're going to come in to the center portion or the medial portion of that eye there, and you're going to find that lacrimal punctum. And then it comes into the lacrimal caniculus, which is going to be a tiny little tube that's going to lead to the nasolacrimal duct. That nasolacrimal duct is going to be a slightly larger tube, which is open, that's going to lead into the nose. And from there, it's going to be able to drain out through that nostril. Now here you're looking at those different muscles. So remember, in with the different muscles as you're looking at them, that's going to help to control and move that eye around. So starting from the top, you have that superior oblique. Remember, that's going to rotate the eye downward and away from the midline. Underneath, you have that superior oblique tendon, which is going to also help to move that eye around. You have that superior rectus, and that's going to help to move that eye and rotate it upward towards that midline. And you have the lateral rectus muscle, which is going to be right below that, and that's going to rotate the eye away from the midline. Now coming down, let's go across starting from left to right. You have the inferior rectus muscle. That's going to rotate the eye upward and away from the midline. And you have the inferior oblique muscle. And that rotates the eye upward. Sorry, that one rotates the eye upward and away from the midline. The lateral rectus rotates the eye away from the midline. So those are going to help to control that eye. And here is another view, looking at it coming in from the inner portion or deep within that eye socket or the orbital. Notice how they all come together at a common tendinous ring, which is going to put them together. And then you're going to see the optic nerve, which will be towards the back. Now, the optic nerve is going to be attached to the back portion of the eyeball where that sclera is. You cannot see it because of that superior rectus muscle going across. But if you were to snip that, move that away, you would see that yellow cord is actually attached into the back of the eye right there. And from the inside of the eye, it will actually create a blind spot, which we'll look at in a little bit. And so here's just a breakdown of those movements of the eye muscles and the controlling cranial nerve. Now going into the eye, the different portions we're going to go over, you have your fibrous layers, which are going to be the cornea and the sclera. These are going to be made up of the same type of tough connective tissue, but the cornea is going to have all of its fibers going in the same direction. This is going to cause it to be see-through and clear. The sclera is actually going to be made up of these same fibers, except for these fibers are going to be overlapping, some parallel, some horizontal. They're going every which ways, and this makes it so they are not see-through. So it creates a very tough white outer coating or tough exterior, whereas the cornea is completely see-through, so you cannot, or so you can see everything through it. So one's clear, one is not. Now coming down to the inner layer, you're going to have that retina layer there. And so that's going to be the layer that's going to contain your cones and rods. Those are going to be the cells that are going to be able to pick up on the information that's coming through the pupil. And so as that information comes through, it is going to be projected onto that neural layer of the retina. And that neural layer is going to be able to take that information using the cones and rods and send it through the optic nerve, which is going to be this center portion right here and send that to the brain. So we have that neural layer. Underneath that neural layer, you're going to have a pigmented layer there. That pigmented layer is going to be referred to as the choroid process. So we have that pigmented layer, and then you're going to have the choroid, which is right under that pigmented layer. That you're going to be able to find different things in it, such as the blood vessels and things that are going to feed that eye. Coming further down, you're going to have that tough sclera, which is going to be continuous of that white, tough, connective fibrous tissue that you saw in the picture that we were looking at. Coming over to the right-hand side, you see the iris. That is going to be the colored part of the eye. It's going to be controlled by smooth muscle. Underneath, you're going to have the lens, which is going to be this portion here. The lens can plump up, which will help you see things close together, or it can be stretched, and it's going to be stretched by these muscles here called ciliary bodies, and that's going to help you see things further away. So this lens here is able to stretch, or it's able to plump itself up, depending on what it's trying to look at. When the light passes through the lens, it's going to be projected onto this back layer here, which is going to be that retinal layer. And then all that information will be picked up and sent through this optic nerve to the brain to be processed. Now, this one small area right here where that optic nerve is going to be referred to as the blind spot, because there are no cells there that can pick up on shading or color or any type of information. So that will be a blind spot. So with the structure of the eye, it's going to be a hollow spherical structure about 2.5 centimeters in diameter. It's going to have a wall that has 3 distinct layers: an outer fibrous unit, which is going to also come together with a middle vascular tunic, and then you're going to have an inner nervous tunic. And so the spaces in the eye are also going to be filled with fluids that are going to help to maintain the eye's shape. With the cornea, that's going to be a clear portion that's going to allow light into the eye. The sclera is the white portion of the eye. Remember, we said that was a tough, fibrous connective tissue. And then going into that vascular layer, you're going to have the choroid, which is where you're going to find the nutrient-rich or blood vessel-rich layer. That's going to be the layer that's going to be feeding the eye. So it has a lot of melanocytes there to produce melanin. That's going to help to absorb any excess light that's going to be found inside of the eye. You have ciliary bodies. That's a group of ciliary processes and muscles that's able to form a circular round, a circle that's going to go around the lens. So that's going to be controlled by smooth muscle, and it is not under your voluntary control. And last, you have the lens, and we talked about it being able to plump itself up or to stretch. Now, that's pretty much going to be a clear lens. Now, as you get older, sometimes it does start to become opaque. It can form cataracts over it. So we'll talk about that in a little bit. So here we are looking at it just a little bit closer. You can see you got your ciliary processes of the ciliary body. You've got those suspensory ligaments, and that's what's going to be actually attached to that lens. So the ciliary processes attached to the muscle are going to be able to pull using those suspensory ligaments, and that's what's able to stretch that lens out or to plump it up. You see the retina. That's going to be the part of the eye that's got all of the cells in it that's able to process and gather up that light information. You've got the choroid. Remember, that's going to be the vascular layer that's feeding the eye. And then the outermost layer, which is going to be the protective layer, is that white section called the sclera. So here in this diagram, it's a great visual of what's going on within the eye. It kind of gives you a show of almost everything that you're going to need to know. So first, we're coming in with these ciliary bodies. These ciliary bodies, remember, are going to be muscles that are able to contract or relax. They're going to be attached to these suspensory ligaments, which can also be referred to as ciliary zonules. They're attached to the lens. So remember, the lens is going to be made up of, it's going to be a protein of crystalline substances which turn into fibers, and as these fibers start to all develop in the same direction, it's going to be able to focus this light that's coming through the pupil. And so next, when we're coming down, we have the cornea, which is going to be that clear section of the eye. All those fibers are going in the same direction. Once it hits the sclera down here, then they're kind of going every which ways. The iris portion that you see here is going to be that colored portion. Now, with this, some people have melanocytes that are growing over top, and that's going to create different colors of the eye. Everybody has blue eyes. But some people will have an extra layer of melanocytes that start to grow over top of this iris portion. Depending on the color of melanin they have, and depending on how much they have, depends on what color eyes they have. For example, I have brown, so with my eyes, I have a layer of melanocytes that's producing a dark melanin that's growing over top of my regular iris. And so with that, it kind of acts like a pair of sunglasses. It helps to not scatter the light. It helps me not to be as light sensitive. And it's going to help me when I'm outside in very bright conditions be able to see better. Now, people that do not have those melanocytes will be more sensitive to light. All right, coming on down, you have the pupil. The pupil is simply going to be a hole. It's an opening that's going to allow light to come through. So the light will enter through the cornea, come through a liquid portion, which is going to be found here. It's called the aqueous humor. So it's kind of a clear, liquid, watery substance. Comes through that, through the pupil, passes across or through that lens. And when it's going through the lens, that lens could either be stretched so you can see because you're trying to look at something far away, or it could be relaxed and kind of plumped up if you're trying to read something close up. And so that's going to help to focus, that lens does. And when it focuses, it's going to focus on this back portion here that you're going to see, which is referred to as the retina. So the retina is going to be the actual layer that's helping to gather this light, process this light through cells that are going to be your rods and cones. Inside of the eye, you're going to have another liquidy substance. It's more jelly-like, though. If you were to do an eye dissection in class, it's going to be very resemblant of Jell-O, but a little bit more liquidy in substance. And so that's going to be called the vitreous humor. And so that's what you see there. That's going to help keep the eye shape, and it's going to help to keep the pressure proper within inside the eye. Now coming over, we have on the right-hand side, we have that continuous white section, which is going to be the sclera. You have your choroid layer, which you can see all those vessels going through. That's going to be the vascular layer. You have the retina, which we were talking about. That's going to be the yellow layer that has all of the visual cells in it. Then you have what's referred to as that fovea centralis. And then coming down, you're going to have where the optic nerve comes in. The fovea centralis is going to be where they're attached in to like that choroid area. And then when you come down to the optic nerve, you'll have your optic disc, or what we refer to as the blind spot, right through here. And that's going to be a section that you can't see. Also coming in with the optic nerve are going to be your central artery and central veins of the retina that are coming in to feed those cells. Now with vision, you can have different types of refraction. So when a person sees an object, either the object's giving off light or it's reflecting light from another source. So rays of light are going to enter into the eye, and the image of what's being seen focuses upon the retina. The light rays must bend to focus. It's a phenomena which we call refraction. So focusing bends the light so that the image falls on the fovea centralis. So that's going to be the portion where your light is going to have that focal plane. So you can see that here. So with emmetropic eyes, these are going to be normal vision. These are going to be the people who their focal point is perfect and they can see. This is 20/20 vision. So this refraction is going to occur when light rays pass at an angle from a medium from one density into a medium of a different density. For example, when light passes from the lens less dense medium such as air into a denser medium such as glass, or from air into the cornea of the eye, the light's going to bend towards the line perpendicular to the surface between these substances. So that light's going to bend again when it re-enters the air. So when the surface between such refraction media is going to be curved, that lens is going to be formed. And so a lens with a convex surface is going to cause those light waves to converge, and the lens with the concave surface is going to cause the wavelights to diverge. And so we're going to look at this. This is, of course, normal vision. Going into the next one, you can see the myopic eye. That's going to be people who are nearsighted. For example, I am nearsighted. And so what's happening here is that the eye is going to be too long. And so when it comes in to focus the point, that image is going to focus before it gets to that retina or that fovea australis back there. So when you look at that, notice that it's not focusing on the retina. Instead, it's focusing too soon. And so that light is going to scatter and it's going to cause things to be blurry if it's uncorrected. And so your focal point is going to be kind of towards the middle of the eye where you're not going to have those cells. So it's going to be blurry when it does focus. Now, when it's corrected using a concave lens, that's going to move that focal point further back and it's going to focus on the correct portion, and then the eye is going to be able to sense and send the proper signal to the brain so you can see clearly. Hyperopic eye, or farsightedness, is going to focus behind the eye. So that's when the eyeball is going to be too short. So that focal point is behind the retina. With this, you go in with a convex corrective lens, and that's going to move the focal point closer in, and then it's going to focus on that retina, so you're able to produce a clear image. So with this, the ciliary muscle fibers are actually going to help you to focus this on the back of the eye, on the retina. And so with this, those ciliary muscle fibers relaxed, and you have those sensory ligaments taut, and that lens is going to be thin. So you're trying to see something far away here with this top picture. So that's a distance view. When you're coming down to the next picture, it shows you how it's plumped up that lens. It's made it thicker. That's when you're looking at something that's going to be closer together. So each of these is going to use different parts of the nervous system. The top one, when you're using distance, is going to be sympathetic nervous system. When you're looking at things closer up, it's going to use the parasympathetic nervous system. Another thing that occurs as images go through a lens. If you've ever used a microscope, you might have noticed this. In lots of labs I try when we first start out in anatomy as well as biology, I try to get students to notice this. If you put something under the microscope where it's going through lenses, you're going to notice that it's going to do two things. The image is going to flip upside down, but it's also going to reverse itself. So it flips and reverses. And so if you move a slide and you think you're moving it towards yourself when you're looking at it, if you're looking through the actual objective lens, it looks like it's moving away from you. If the E is facing you, and it's where you can see it when you put it onto the stage of the microscope, when you look through the lens, it's upside down now. But it's not just upside down, it's also flipped in reverse. Right. So here we have sympathetic activation. So here, the lens, you're trying to see distance. It's going to flatten that lens out. So sympathetic input is going to relax those ciliary muscles. It tightens the ciliary zonules, which are these ligaments, and it helps to flatten the lens. Notice when you're looking at it, how it flips and reverses. Parasympathetic, this is going to help to plump up that lens. So your parasympathetic input is going to contract those ciliary muscles. It loosens the ciliary zonule or ligaments, and it allows that lens to bulge. Still, we're flipping and inverting it. Now going into the muscles for this. With this parasympathetic, it's going to use sphincter papillae muscle contractions, and it's going to cause the pupil size to decrease. The iris is going to have two muscles embedded within it. It's going to have a sphincter papillae, and any type of sphincter muscle that you learn is going to be a circular muscle, and you're going to have a dilator papillae muscle. The sympathetic nervous system is going to activate that dilator papilla muscle and cause it to contract, and that's going to cause the pupil size to increase. So here we notice that the iris is the colored portion of the eye. And so it consists, like we looked at the pictures, both of smooth muscle, and it's going to have a hole in the center called a pupil. Now nothing's in that hole, it's just an opening. Going into the inner layer of the eye, the retina is going to be that pigmented layer that's going to help to absorb light. It has a neural layer in it that's going to contain your photoreceptors, your bipolar cells, and your ganglion cells. The fovea centralis is going to be the portion of the retina with the greatest concentration of photoreceptors. So this is going to give you the sharpest vision at that point. You have the optic disc, which is going to be the portion of the retina that is going to lack photoreceptors, and that's going to be referred to as your blind spot. Remember, that's where the blood vessels are coming in and out, and that's where you're going to have your optic nerve. And so looking at the picture here, this is simply going to show you these different sections. So we have our optic disc, which is your blind spot. You can see that neural layer. You have your pigmented layer behind it. And then you have what's going to be that optic nerve, which contains the central vein and the central artery. So on the screen here, you're going to have a blind spot test. Here, you're going to see an image with a plus sign on one side and a dot on the other. So what you're going to have to do is close one eye. Typically, you're going to start with the left eye closed, and you're going to focus on the plus sign with your open right eye. Then slowly move your head or put a paper in front of the screen and for a closer or let's say farther until the dot appears or disappears from your view. So at a certain point, the dot is going to start to vanish. This is when it's going to fall in your blind spot. So despite the dot disappearing, your brain's actually going to fill in this area using the surrounding background. And so this is going to show you how your perception's constructed, not just passively as it's received. And so really, this is going to be important because it's going to demonstrate that we all have a blind spot, but we rarely notice it due to how the brain's able to come in and compensate and fill in that space. So it's a fundamental concept in vision science and neurology. So this is going to be used in both things such as education and diagnostics to see what your vision is and find out what your blind spot is. So in this diagram, it's going to focus in on the complex structure of that retina. It's going to have distinct layers. It's going to have retinal pigment epithelium. It has neurons, nerve fibers, and it's going to have those limiting membranes there. So there's going to be 5 major groups of retinal neurons. The nerve fibers of the 3 major groups are going to be the photoreceptors, the bipolar neurons, and the ganglion cells. These are going to provide a direct pathway for impulses triggered in the photoreceptors to the optic nerve and brain. The nerve fibers of the outer 2 groups are going to be the retinal cells called the horizontal cells and the amacrine cells. These are going to pass laterally between the retinal cells. So the horizontal and amacrine cells are going to modify the pattern of impulses that are conducted on the fibers in that direct pathway. So as you see, the light is coming in through this vitreous humor. Remember, that's going to be that thick jelly-like liquidy substance. We have all of these nerve fibers, which are connecting in with these neurons. So it's going to come in, go through those. We have the ganglion cells, which are connected to the amacrine cells, which are also the ganglions connected to some of the bipolar cells. They come in, you have your horizontal cells, and you can see how they're all connected in with the different rods and cones. This is going to be a microscopic view of it, so you can look at this photomicrograph of the retina and see what these cells actually look like in these different layers. Now coming in, you can look at the rods and the cones. They have specific shapes. Rods are going to be thinner and longer. Rods are also going to have projections at their terminal ends. Now the others that you see there, the cones, are going to have short, blunt projections. So the retina has about 100 million rods and about 3 million cones. Rods and cones are going to occupy that deep layer of the retina that's going to be closely associated with a layer of retinal pigmented epithelium. the projections from the receptors are going to extend into that pigmented layer, then contain light-sensitive visual pigments. So retinal pigment epithelium is going to help to absorb that light in those receptor cells that they don't absorb. And with that, the pigment in the choroid coat is going to keep the light from reflecting off the surface of the inside of the eye. So that's going to be important. That retinal pigmented epithelium also is going to help to store things like vitamin A. Remember, vitamin A comes from things like carrots. And so that's why they say eat a lot of carrots in order to have healthy eyes, because it helps to nourish that portion of the eyes. So rods and cones are going to work in different ways. Rods are going to be 100 times more sensitive to light than cones are. So rods provide vision that's going to be good in dim light. Also, rods produce vision without color. So these are going to be in shades of gray, where cones are going to be able to detect specific colors. Cones, however, are going to be less sensitive and they don't respond to dim light. So cones provide sharp images, whereas rods produce a more general outline of objects. That's going to be because the nerve fibers associated with many rods may converge, and so those impulses may be conducted to the brain in one single nerve fiber, whereas with the light that's going to stimulate, when it gets to the brain, it can't tell which one of these receptors has been stimulated. So it kind of goes in, this is going to create like peripheral vision, it creates shades, and it's good for dim light. The sharpest vision, like we said, and the color is going to actually come from the cones. And so that's going to come in. And remember, your sharpest vision is going to be found specifically at that fovea centralis. That area is actually going to lack rods, and so it's only going to have cones in that area. So it's going to be densely packed with those, and it has those converging fibers. Here you can see the fovea centralis, so that's going to be that area of the sharpest vision. And over to the side, you see that blind spot, or the optic disc. Now, sometimes when you go to the eye doctor, they'll use what looks like a glass. They'll shine light through it, and they look into your eye. This is some of the things that they're looking for. Here you see retinal detachment. That's where that retina is starting to detach from the back portion of the eye. And you can also see if there's a retinal tear. Now, if you have a retinal detachment, you will actually start to lose your vision. You might, as it's detaching, start to see sparks of bright white. It might look like sparkles or flashes. When it completely detaches, it all becomes white. You might think it would be dark, but it actually just becomes like a bright white, they say. Here's some different things that you can see in this one. Of course, this eye is in bad shape. It's got some hemorrhages, so that's where the blood vessels have ruptured. You can see the arterioles and the venules. You see that optic disc. You see exudates. That's going to be portions of detachment there. And you see microaneurysms within this eye. So chambers, fluids, and other internal parts. So the anterior chamber is going to be the portion that's going to be in front of the lens, but behind the cornea. And so this fluid is going to help to nourish and oxygenate the cornea and the lens. So this one's more of a liquidy. The posterior chamber, remember, contains that vitreous humor. That's that jelly-like fluid that's going to help to support the internal eye, and it's going to help to maintain its shape. It also helps to maintain the pressure. So sometimes they'll blow that light into your eye. That's glaucoma. That's checking for how your eye responds to pressure. And so looking at this, you can see that cornea, you see the pupil, you see the iris, that anterior chambers where you're going to find that aqueous humor, and behind it, that posterior cavity is where you're going to find that vitreous humor. Now with this, notice how you do have fluids that can come across the eye, and you can see this as it's coming across that picture, and it comes out that scleral venous sinus there. So I like this diagram or this picture because it shows you what or how the light's being gathered and taken in through the eye to the different parts of the brain. So you have your specific fixation point, and they're going to use two different colors so you can see how it travels through these different fibers to the different parts of the brain. So coming in, let's start on the left-hand side, which of course would be the right because we're in anatomy. And so it's coming in, let's say, to the right eye. Anything in blue you see coming in through those blue fibers, as well as the left eye. Notice how you're picking up from the side and it's coming in through those blue fibers. Notice how the clearer portion is going to come through that optic nerve and cross through that optic chiasma. And once it hits the optic chiasma, that's where it crosses over to the opposite side of the brain using the optic tract. The left side, you're picking up yellow, but notice how it also picks up on the lateral portion of that right eye. It comes in, crosses over to the right side of the brain, and at that optic chiasma and follows that optic tract. So basically, it's going to include the optic nerve, the optic chiasma, the optic tract, and optic radiations that you see coming down through. So these nerve fibers are going to continue in the optic tracts, and before they reach the thalamus, a few of them are going to enter into the nuclei that function in various visual reflexes. Most of the fibers, however, are going to enter the thalamus and synapse in a part of its posterior portion, which is going to be that lateral ganglia body. Geniculate of that nucleus of the thalamus. So from this region, the visual sensory fibers are going to enter into pathways called optic radiations, and that's going to lead them to the visual cortex of the left and right occipital lobes. So in this picture, it actually shows you what it looks like in a cadaver brain. So you're able to visualize how you have your optic chiasma crossing over. It's coming in from the optic nerve, crosses over the optic chiasma, goes into the lateral geniculate nucleus, and then travels out through the optic radiation to the occipital part of the brain. All right, so that leads us into our last sense that we're going to discuss, and that's going to be the auditory sense. So this is our sense of hearing. So the organ of hearing is going to be the ear. It's going to have the outer external portion, the middle portion, and the inner or internal sections. In addition, it's going to make hearing possible. The ear is going to help to provide a sense of equilibrium, so a sense of movement if you're standing up, if you're laying down, or if you're upside down. So let's start out with the outer ear portion. The outer ear is going to consist of all the structures that face the outside. These include the outer funnel-like structure called the auricle, or sometimes referred to as the pinna. It has an S-shaped tube, the external acoustic meatus, and it's going to have that internal auditory canal. And as you see that, that's actually going to go in through that temporal bone. And so when you just studied the bones, you would have known that as the external acoustic meatus. Now, the external acoustic meatus passes through that bone. Near the opening, it's also going to have hairs that are going to help to guard this tube. The opening and the tube are going to be lined with the skin that has many modified sweat glands called ceruminous glands, and these are going to help to secrete cerumen, or what we call earwax. The hairs in the wax are going to help to keep the large foreign objects such as insects and things like that out of our ears. So major parts of the ear that you're going to see here, of course, are going to be that outer portion, and that's your auricle. It's got different parts to it. You have that helix, you have the lobe, which people like to pierce. Notice how that lobe is full of a lot of adipose tissue. Notice how the outer portion also is formed with cartilage, and that's going to be elastic cartilage. You come in through the tunnel, which is that external acoustic meatus. Next, you're going to enter into the middle ear. And so with the middle ear, that's going to also be referred to as the tympanic cavity. That basically is going to be an air-filled space in the temporal bone that's going to help to separate the outer and inner ears. It's going to be bound by that tympanic membrane, which is actually the eardrum, which you would know as the eardrum. And inside there, you're going to have auditory ossicles that are connected to the eardrum or the tympanic membrane. Now with those, it's going to be 3 bones. These are going to be the smallest bones found in the body. They're called the malleus, the incus, and the stapes. And they're going to be attached to the wall of that tympanic cavity and tiny little ligaments that are going to be in there. And they're covered by a mucous membrane. These bones are going to bridge that tympanic membrane with the inner ear. So it's going to help to transfer vibrations between these parts. Specifically, that malleus is going to be attached to the tympanic membrane, and it's going to help to maintain its conical shape. So it makes it kind of like a cone. When the tympanic membrane vibrates, that malleus is then going to vibrate in unison with it. It's going to send that vibration into the incus, And then from the incus, it's going to pass it to the stapes. There, you're going to have an oval ligament that's going to hold the stapes to an opening in the wall of that tympanic cavity called the oval window. And so we'll look at that here. And so you can see that oval window that it's attached to. So that's where that stapes is attached. So vibrations from that stapes, which is going to act like a position of the oval window, it's going to transfer those vibrations into a fluid within that inner ear. So those vibrations with that fluid are going to help to stimulate those hearing receptors. So below those ossicles, you're going to see the round window. It's going to be a thin, flexible membrane that's going to be able to form a boundary between the middle ear and the fluid-filled cochlea of the inner ear. And so its main job is to act basically as a pressure release valve for the waves that are going to be generated in that cochlear fluid. So here's what happens. So the sound waves are going to enter from that ear canal. That vibration is going to go through that tympanic membrane, which remember is your eardrum. It's going to pass through the ossicles, which are the malleus, the incus, and the stapes. Then from that stapes, it's going to go in and it pushes that oval window. That's going to begin to transmit those vibrations into that paralymph fluid inside of the cochlea, which we're getting ready to look at. So because that fluid is going to be incompressible, that wave is going to need an outlet. And so this is where that round window comes in. The round window is going to bulge kind of outwards, and the pressure waves are going to travel through the cochlea because of this. And so the movement of fluid inside the cochlea is going to cause the basilar membrane to move. That's going to stimulate the hairs, which then convert mechanical energy into nerve impulses, and then the brain's going to interpret that as sound. So the inner ear is going to be made up of semicircular canals. The function of these is going to be for equilibrium. You have the cochlea. The function of the cochlea is going to be for hearing. Both parts are going to be composed of a series of tubes. So looking here, we see our bony labyrinth of the semicircular canals. It's going to have that outer portion, which is then going to be lined with an inner portion, which is going to be called the membranous labyrinth. That's going to contain the endolymph. And then all of those together, all those different circles are called the semicircular canals. Notice how you'll have a nerve that comes in and it will attach to the main area there called the maculae. And then it also has that round window that we talked about where that pressure is able to expand and compress and send those sound waves through. The cochlear nerve is going to come in, and the cochlea is actually this little squiggly part that kind of looks like a snail shell, and it's going to come in and attach to that. Notice how they will come together and make a vestibulocochlear nerve. So here we're going to focus in on the cochlea. The cochlea is going to be a tube that's going to be widest at its base and progressively narrower as it gets towards its tip or its apex. It's going to be a bit shaped like a snail shell. It's going to be coiled around a bony core. It's going to have a thin, bony shelf and extend out from the core and coils around within the tube. So a portion of the membranous labyrinth in the cochlea is going to be called the cochlear duct or the scala media. That's going to run inside the tube opposite the spiral lamina, and together these structures are going to divide the tube into upper and lower compartments. So within that upper compartment, it's going to be called the scala vestibula. It's going to lead to an oval window to the apex of that spiral. The lower compartment, the scala tympani, is going to extend from the apex of the spiral to the membrane-covered opening in the wall of the middle ear, and that's facing that tympanic cavity called the round window. And so those compartments from the bony labyrinths of the cochlea are going to be filled with a paralymph. The cochlear duct between it is going to be filled with an endolymph. So at the apex of the cochlea, beyond the tip of the cochlear duct, a small opening, which is going to be called the helicotrema, is going to connect the paralymph in the upper and lower compartments, and it's going to allow that fluid pressure in them to equalize. So you have that cochlear duct that's going to end as a closed sac at the apex of the cochlea, if you can see that there. Let's move on to the next picture. So here you can see that cochlear duct a little bit better and those different membranes there. So here, looking at that basilar membrane, it's going to extend from that bony shelf that you see of the cochlea, and it's going to form a floor of the cochlear duct. It's going to have many thousands of elastic fibers that are going through it. So vibrations that are entering in through that perilymph at the oval window are going to travel along the scala vestibuli, and they're going to pass through the vestibular membrane to enter into that endolymph of that cochlear duct. That's where it's going to move the basilar membrane. After passing through that basilar membrane, the vibrations are going to enter into the perilymph of the scala tympani, and that movement from that membrane covering the round window is going to dissipate their force into the air and the tympanic cavity. So looking here, you see that you have the spiral ganglion. That's going to be the section. You see those nerve fibers coming into that spiral organ. The spiral organ is going to contain the hearing receptor cells, and that's going to be on that superior surface of the basal membrane, and it's going to stretch from the apex of that cochlea to the base of the cochlea. And so the receptor cells there are going to be called hair cells. So this is looking at it a little bit closer. And so we have our many hair cells. They're going to have processes known as stereovoli or stereocilia. That's going to extend into the endolymph of the cochlear duct. So above these hair cells is going to be a membrane that's going to attach to the bony shelf of the cochlea, and that's going to extend like an overhang over top of the hairs of those cells. And so you see those supporting hair cells, and then you see the stereocilia coming out and that tinctorial membrane that's going to be above it that we just talked about. So with these hair cells, they're going to be arranged in two groups. You have a single row of about 3,500 inner hair cells and 3 rows of about 15,000 to 20,000 outer hair cells. So it's going to be the inner hair cells that's going to be the actual hearing receptors. So as sound vibrations cause movement of that basilar membrane, some of those inner hair cells are going to be pushed against that tinctorial membrane, and it's going to bend those hairs. So in response to the hair cells being stimulated by those impulses on the fibers of that cochlear nerve, it's going to send that information and reach those auditory centers in that temporal lobe, and that's going to be interpreted as hearing. So the outer cells that you're going to see are going to make the mechanism more sensitive to the lower sound volumes. So those stereocilia of those outer hair cells are going to be anchored to the tinctorial membrane above them. So in response, even very low volume sounds, they're going to be able to pull down that membrane, even when it's super low, and bring it closer to the stereocilia in the inner hair cells, and it's going to make the stimulation of the inner hair cell more likely. And so that causes that inner hair cell to be more responsive to very quiet sounds. Now, that basilar membrane is going to be stiffer and narrower at the base of the cochlear compared to the apex. And so it's going to be more flexible and wider there at the before it becomes narrower. As a result, this is going to pick up on different frequencies of vibration and move different regions along the length of that basilar membrane. So a particular sound frequency is going to bend the hairs in a specific group of those receptor cells, and that's going to activate them. Other frequencies are going to activate receptor cells elsewhere. And so along that cochlea, it might pick up different frequencies, and that's how that sound's going to activate those receptor cells in different places along that basilar membrane, different times and simultaneously. And that's how you're going to be able to hear multiple tones at the same time. And so here shows you how it's going to be coming in. So you see your sound waves coming in. They vibrate that tympanic membrane. Then it goes to the auditory ossicles to vibrate, and pressure is going to be amplified. Then the pressure waves are going to be created by the stapes pushing on that oval window to move through the scala vestibuli. Then the sound with frequencies below hearing are going to travel through the helictotremia and then excite those hair cells. And then sounds in the hearing range that go through the cochlear duct are going to vibrate that basilar membrane and deflect the hairs on the inner hair cells. And then you see how it's attached with that cochlear nerve. And this simply shows you how it moves. So high frequency sounds are going to displace the basilar membrane near the base. Medium are going to displace the basilar membrane near the middle. And low frequencies will displace the basilar membrane near the apex or the end. The physiology of hearing, here's the different steps. So the sound waves are going to enter external acoustic meatus and cause the tympanic membrane to vibrate. We've discussed this. The malleus, incus, and stapes are going to transmit those vibrations to the oval window. That oval window transmits the sound waves via the perilymph of the scala vestibuli to the vestibular membranes. Sound is going to be transmitted from the vestibular membrane to the endolymph of the cochlear duct. These are going to cause that basilar membrane to vibrate. This vibration is going to move hair cells in the organ of corti to move back and forth. Hair cells are going to stimulate the nerve cells to transmit information to the brain via the cochlear nerve. And then the leftover waves are going to be transmitted into the perilymph of the scala tympani, which is going to be dissipated into the tympanic cavity via the round window. So the cochlear branches of the vestibulocochlear nerves are going to enter the auditory nerve pathway that extend into the medulla oblongata and proceed through the midbrain to the thalamus. From there, they're going to pass into the auditory cortices of the temporal lobes and the cerebrum, where they're going to be interpreted. So on the way, some of these fibers are going to cross over so that the impulses that are coming from each ear are going to be interpreted on both sides of the brain. Consequently, damage to the temporal lobe on one side of the brain is not necessarily going to be accompanied by complete hearing loss in the ear on that side. So the pathway of vibrations through parts of the middle and inner ears is going to come up, and you can see how it enters into that cochlear nuclei there. And from there, it comes through that section of the medulla, which is going to be that superior olivary nucleus, or the pons medulla junction. It's going to come up through the midbrain. It's going to enter into that primary auditory cortex of the temporal lobe, and then you see it go through that medial area and then come out into the temporal lobe. Equilibrium. So equilibrium is going to be your sense of balance, your sense of direction, what you're doing. Static equilibrium is going to be a sense of position of the head. It's going to help to maintain posture and stability when the head and body are going to be still. The vestibule is going to be composed of two chambers called the utricle and the saccule. Each of these chambers is going to have tiny structures called maculae. And so looking here, these semicircular canals are going to be what you are going to use in order to determine your direction, if you've been moving, and what your position of your body are. So the maculae here are going to respond to changes in hip positions. So maculae on the utricle with the head in an upright position and maculae of the utricle with the head in the bent forward position are going to change how those different hair cells are going to respond. So to the next one. So you see the hair cells here. That's going to have those stereocilia on the end. Notice that you have the otolith membrane, and then you have otoliths. Otoliths are going to be little stones that are going to be in this gel-like membranous fluid. And so as you bend your head, it's going to cause these hairs on these cells to bend with it. So it can tell if you're upright, or if you bend your head down, for example, it's going to be able to move those. And those otoliths also in that gelatinous material are going to move. And that's going to help you to send signals to your brain to let it know that your head is facing down. Dynamic equilibrium is going to detect motion when the head and body move. So this information is going to help the brain in maintaining balance. So the semicircular canals are going to be the organs of equilibrium, like I said. They're associated with each semicircular canal as a swelling called an ampulla. So each ampulla is going to contain a sensory organ called the crista ampullaris, and that's going to bend with the motion of the head. And so that sends signals to the brain about the motion of the body. So in this picture, it's going to show you the crista ampullaris and how it's going to be, or where it's going to be found in each semicircular duct. So the ampulla is going to contain a ridge that's going to cross the tube that houses the sensory organ. And so it's called the crista ampullaris. It's going to have a number of these sensory hairs that you see inside and supporting cells. So as the maculae, the hairs, and the hair cells extend upward into a dome-shaped gelatinous mass called that ampulla, or cupula, sorry, the hair cells of the crista ampullaris are going to be connected at their bases to dendrites of neurons, and that's going to make up part of the vestibular branch of that vestibulocochlear nerve. And so when the head or torso moves, those semicircular canals are going to move as well. But initially, that fluid that's going to be inside the membranous ducts tends to remain stationary because of inertia. So the bending of that copula is going to be in one or more of the semicircular canals is going to be the direction opposite that with the head and torso is going to be actually moving. And so those hairs that are embedded also are going to start to bend. And the moving of those hairs is going to stimulate the hair cells to signal to their associated neurons, and that's going to result in an impulse that's going to then be conducted into the brain. So when the head is stationary, the capula of the crista ampullaris is going to remain upright, but when the head's moving rapidly, you're going to see that capula bend opposite the motion of the head, and it's going to stimulate those nerve receptors. So information about the body's position in space is going to come from three main sources, and it's going to be fed into two major processing areas in the central nervous system. So first one we're going to look at is those vestibular receptors. It's going to come in and it's going to process at the cerebellum, but also notice it could cross over and process at the vestibular nuclei, and that's going to be inside the brainstem. When it goes to the cerebellum, it then can go to the vestibular nuclei as well and come down, and you have your ocular motor control, and that's going to be done through the cranial nerves, nuclei 3, 4, and then 6. Visual receptors come in, and you can see how they can go into the cerebellum or the vestibular nuclei in the brainstem, and then you can also see your somatic receptors coming in as well, the skin, muscle, and joints. And so with those, notice how you can go into the ocular motor control, or it could go into that spinal motor control through those different cranial nerves. So the output is going to be your responses by the central nervous system that provide fast reflexive control and muscle serving, which are going to be the muscle serving, the eyes, the neck, the limbs, and the trunk. And that's going to finish us up for our senses. So this is our last unit for Anatomy 141 or Anatomy 2. I hope you enjoyed this course, and hopefully we'll see you for Anatomy 2.